Okay, let's unpack this. Imagine a blueprint, right?
Hmm.
Thousands of pages long, all filled with these coded messages.
Hmm.
Now, picture this bustling factory taking that code and precisely assembling incredibly complex machines, one tiny piece at a time, but at lightning speed.
Yeah, that's a great analogy.
That's essentially what our cells are doing right now, tirelessly converting genetic instructions into the machinery of life. And it's truly mind-blowing when you think about it.
It absolutely is. And what's fascinating here is just how universal this process is, yet at the same time, how exquisitely detailed it is.
Right.
Today, our deep dive is all about translation. That's the fundamental biological mechanism that builds proteins from messenger RNA or mRNA.
And we're grounding this discussion.
We are. We're drawing our insights primarily from Chapter 13 of Genetics, Analysis and Principles by Robert J. Brooker, 7th edition.
Okay.
Our mission really is to break down the core mechanisms, look at the experimental breakthroughs that revealed this whole intricate process, and introduce the key molecular players involved.
So by the end of this, you listening should have a really solid grasp of how this genetic code gets turned into functional proteins.
Exactly. You'll leave feeling hopefully truly well-informed.
Okay, so before we dive headfirst into the nitty-gritty of translation itself, maybe let's rewind a bit. How did anyone even first suspect the genes were telling our bodies how to make specific things, especially enzymes, those biological catalysts? What was the first clue?
That's a great starting point. And the story really begins over a century ago with Archibald Garrod, a British physician, a real pioneer.
Garrod, right?
He studied these inherited conditions that he called inborn errors of metabolism. And his most famous work was on alkaptonuria.
Alkaptonuria.
Yeah.
What happens with that?
Well, it's a disorder where patients accumulate this compound called homogentic acid. The signs are actually quite striking.
Oh, yeah.
Their urine turns black when it's exposed to air, and over time they can get this sort of bluish-black discoloration in their cartilage and skin.
Wow. Okay. So distinctive signs.
Very. And Garrod realized it was inherited, passed down in what we call an autosomal recessive pattern.
Meaning you need a faulty gene copy from both parents.
Precisely. And he made this brilliant deduction. The buildup of that compound, the homogentic acid, was because of a missing or defective enzyme, specifically homogentic acid oxidase, which normally breaks it down.
Ah, so the enzyme's job wasn't getting done.
Exactly. And this was really the very first concrete link proposed between a specific inherited gene and the actual function of an enzyme, a truly monumental leap in thinking back then.
So Garrett gave us that first hint, genes tied to enzymes. But it was Beadle and Tatum who really pushed it further, asking, is it strictly one gene controlling one enzyme? This is where it gets really interesting, I think.
Spot on. George Beadle and Edward Tatum, working in the early 1940s, wanted to nail down this relationship more precisely. They chose Neurospora crassa. It's a common bread mold as their experimental system.
Why bread mold? Seems a bit random.
Well, Neurospora is actually perfect for this. It's easy to grow in the lab, and it has very simple nutritional needs. It can basically synthesize almost all the essential molecules it needs from a very basic medium.
Okay, so if a gene controls an enzyme for making something essential, they'd look for mutants that suddenly couldn't make it anymore, right?
Yeah.
They'd need it provided in their food.
Precisely their thinking. They reasoned that a mutation, a change in a gene, would knock out the function of one specific enzyme. That would prevent the mold from making some crucial molecule, maybe an amino acid or a vitamin.
And that mutant wouldn't grow on the basic stuff.
Right. It wouldn't grow on the minimal diet. So they irradiated the mold to create mutations, and then they isolated several mutant strains that specifically required the amino acid methionine to grow.
Okay, so they all needed methionine.
Yes, but they hypothesized that each strain had a block at a single different step in the pathway the mold uses to synthesize methionine.
Like different broken steps on an assembly line. So how did they figure out which step was broken in each mutant?
Cleverly, they tested their mutant strains by adding different intermediate compounds, sort of like the partially built components from that assembly line to the minimal media.
Oh,
So imagine strain 1 is blocked right at the beginning. It needs methionine, or any of the intermediate precursors after the block, to grow. Strain 2 might be blocked at step 2, so it could grow if given intermediate 2 or 3 or the final product methionine, but not intermediate 1.
I see. So by seeing which spare parts allowed each mutant to grow.
Exactly. They could meticulously map out the entire biochemical pathway step by step, and they could pinpoint precisely which enzyme, and therefore which gene, was defective in each mutant strain. This led directly to their famous one gene-one enzyme hypothesis.
Groundbreaking stuff.
Yeah.
But science evolves, right? Did that hypothesis hold up perfectly?
It was revolutionary, absolutely. But like many great ideas, it needed some refinement over time.
How so?
Well, first we realized genes encode all kinds of proteins, not just enzymes. Think structural proteins, transport proteins, signaling molecules.
Okay.
Countless roles.
Right. Proteins do way more than just catalyze reactions.
Exactly. Second, many functional proteins, like hemoglobin, which carries oxygen in our blood, are actually made up of multiple different polypeptide chains. Each chain is typically encoded by its own separate gene.
So not one gene for the whole hemoglobin protein, but one gene for each type of chain.
Precisely. So it became more accurate to say one gene, one polypeptide. And third, we discovered that some genes don't even encode polypeptides at all. Their final product is a functional RNA molecule, like transfer RNAs, tRNAs, or ribosomal RNAs, which we'll definitely get into.
Okay.
And just to add another layer, things like alternative splicing mean that even a single gene can sometimes produce multiple different polypeptide variants.
It's never quite as simple as the first elegant hypothesis, is it?
Rarely. But this complexity raises a really important question. Why is it so fundamentally crucial for genetic material to encode proteins? Why are they the main output?
That's a great question. We know genes encode these polypeptides. But how does the cell actually read the instructions from the mRNA molecule to build them? This is where translation really comes in, right? It's like interpreting one language, the nucleotide sequence, into a totally different one, the amino acid sequence.
That's the perfect way to describe it. And the core of this interpretation lies in what we call the genetic code. The mRNA sequence isn't read one letter at a time, but in groups of 3 nucleotides. Each group of 3 is called a codon.
3 letters make a word.
Essentially, yes. Most of these codons are sense codons, meaning they specify a particular amino acid. Like, the codon AGC tells the machinery to add the amino acid serine.
Okay.
Then there's a really important codon, AUG. It does 2 jobs. It specifies the amino acid methionine, and it almost always acts as the start codon, signaling exactly where to begin building the polypeptide.
The starting point. Makes sense.
And just like you need a start signal, you need a stop signal. There are 3 stop codons, UAA, UAG, and UGA. They don't code for any amino acid. They just tell the translation machinery, okay, stop here. The protein is finished.
And you mentioned before there are bits of the mRNA before the start and after the stop that aren't translated.
Yes, exactly. Those are the 5' untranslated region, or 5UTR, before the start codon, and the 3' untranslated region, 3UTR, after the stop codon. They have roles in regulating translation, but they don't become part of the protein.
So how does the cell actually match the right amino acid to the right codon? There must be some kind of go-between.
There is, and these are the fascinating molecules called transfer RNAs, or tRNAs. Each tRNA molecule is like a molecular adapter. It has a specific 3 nucleotide sequence called an anticodon on one end.
Which matches the codon on the mRNA.
Complementary to it, yes, through base pairing. And on the other end of the tRNA, it carries the specific amino acid that corresponds to that mRNA codon. It's the physical link between the nucleic acid language and the protein language.
Like a bilingual dictionary molecule.
There's a brilliant way to put it. Now, this genetic code is incredibly robust. There are 64 possible codons you can make with 4 bases taken 3 at a time, 4 by 4 by 4.
But only 20 common amino acids to code for.
Exactly. So this means the code has degeneracy or redundancy. This isn't a flaw, it's actually a key feature. It means that more than one codon can specify the same amino acid.
Give us an example.
Sure. Take glycine. It can be coded by GGU, GGC, GGA, and GGG. All 4 codons mean add glycine.
So a typo in that third position might not even matter.
Often it doesn't. This redundancy provides a buffer against mutations. A small change in the DNA, particularly in that third codon position, might not change the amino acid sequence at all. It makes the whole system more resilient. And you can see why 3 bases are needed. 2 bases would only give you 16 combinations, 4 by 4, which isn't enough for 20 amino acids. 3 gives you 64, providing plenty of coding capacity and room for this degeneracy.
You mentioned the start codon sets the beginning. This brings up the idea of a reading frame. It sounds technical, but my understanding is it's absolutely critical. Can you break down what a reading frame is and why getting it right is so vital?
It truly is critical. The start codon, usually AUG, establishes this reading frame. Think of it like reading a sentence. If the sentence is, "The fat cat ate the rat," you read it in three-letter chunks.
The fat cat.
Right. But what if you started reading from the second letter? You'd get A, she, A, T, C, A, T, T, her, at. Complete nonsense, right?
Total garbage.
It's the same with mRNA. The ribosome reads the sequence in non-overlapping triplets, starting from the AUG. If you have a sequence, let's say 5RJCC, CCC, GGC, ACC, CACAU3, starting at AUG gives you methionine, then proline, CCC, then glycine, GGA, or so on.
A specific protein sequence.
But if, say, a single base gets accidentally deleted near the beginning, let's say that first C in CCC is lost. Now the sequence reads AUCCG, GAD, GCA. Everything downstream shifts by one base.
Oh, so all the codons after that point are read incorrectly.
Yeah. Completely different triplets. You'd get a totally different sequence of amino acids from that point on, almost certainly resulting in a non-functional, garbled protein, maybe even a harmful one. It really highlights how precise this whole reading process has to be.
And this code is pretty much the same everywhere, isn't it? In bacteria, plants, us.
It's amazing, yes. The genetic code is described as nearly universal. It's largely the same across all known forms of life, which is incredibly strong evidence for a common ancestor for all life on Earth.
Nearly universal. So there are exceptions.
There are a few fascinating ones, mostly minor variations. For example, in the mitochondria of vertebrates like us, the codon AUA codes for methionine instead of isoleucine, which it does in the standard code. And UGA, normally a stop codon, codes for tryptophan in mitochondria.
Interesting quirks.
And even more intriguing are two rare but genetically encoded amino acids, selenocysteine and pyrrolysine, sometimes called the 21st and 22nd amino acids.
How do they get incorporated?
Under specific circumstances, stop codons UGA for selenocysteine, UAG for pyrrolysine can actually be read as coding for these amino acids instead of signaling termination. This usually involves specific sequences or structures in the mRNA downstream of the codon, like a cis element for selenocysteine, that essentially reprogram the ribosome locally.
Wow. The cell even has ways to repurpose stop signals sometimes.
Okay, so these polypeptide chains are built. They have a clear beginning and end, right? It's not just a random jumble.
Absolutely. Think of them as directional chains. The first amino acid added has a free amino group, NH2, and this end is called the amino terminus, or N-terminus.
The start end.
Exactly. And the last amino acid added has a free carboxyl group, -COH, called the carboxyl terminus, or C-terminus, the finish end.
And the links between them? Amino acids are joined together by peptide bonds. This bond forms between the carboxyl group of one amino acid and the amino group of the next, releasing a water molecule in the process, its condensation reaction. And the chain always grows from N-terminus to C-terminus, which directly mirrors the 5 to 3' direction the ribosome reads the mRNA.
So that linear chain, that's just the start of the story for the protein, isn't it? The folding is where the magic happens.
Oh, absolutely. What truly brings these linear chains to life and allows them to perform their incredible array of functions is how they fold into precise, complex, three-dimensional shapes. This folding process happens in stages.
Okay, stage one.
Stage one is the primary structure. That's simply the linear sequence of amino acids itself, dictated directly by the mRNA codons. Remember, each of the 20 common amino acids has a unique side chain, or R group, with different chemical properties. Some are bulky, some small, some oily, hydrophobic, some water-loving, hydrophilic, some charged.
So that sequence is the absolute foundation. What happens next? How does it start folding?
That linear sequence then begins to form localized folding patterns, creating the secondary structure. The two most common types are the alpha helix, all helix, like a spiral staircase, and the beta sheet, bat sheet, which looks like folded pleats.
What holds those shapes together?
These are stabilized mainly by hydrogen bonds within the polypeptide backbone itself, not involving the side chains yet. Think of them as regular repeating structures formed by the chain folding back on itself.
Okay, helices and sheets. Then what?
Then these secondary structure elements, along with the loops and turns connecting them, fold further into a specific compact three-dimensional shape. This is the tertiary structure. It's the overall 3D conformation of a single polypeptide chain.
And this is driven by the side chains.
Exactly. This intricate folding is driven largely by interactions between those diverse amino acid side chains. Hydrophobic side chains tend to bury themselves inside the protein away from water. Charged groups might form ionic bonds, polar groups form hydrogen bonds, and there are weaker van der Waals interactions too. It's a complex interplay aiming for the most stable shape. And often, special helper proteins called chaperones assist in this folding process, preventing mistakes or aggregation.
It sounds incredibly complex to get right, and some proteins are even more complex than that.
Yes, indeed. Many functional proteins aren't just one folded polypeptide. They consist of multiple polypeptide chains called subunits associating together in a specific arrangement. This is the quaternary structure.
Can you give an example?
Hemoglobin is the classic example. It's made of 4 polypeptide subunits, 2 alpha-globin chains, and 2 beta-globin chains, all fitting together precisely to form the functional oxygen-carrying molecule.
So if we connect this all back to the bigger picture, this incredible hierarchical complexity in protein structure from primary sequence right up to quaternary assembly is precisely what enables them to do so many different things in our cells, right? Their specific shape dictates their specific function.
Absolutely spot on. Proteins are the true workhorses and building blocks of the cell. Their incredibly diverse and precise 3D structures allow them to perform a staggering array of functions.
Like what? Give us a few examples.
Oh, sure. Think of tubulin, forming microtubules that give cells shape and act like highways for transport. Hemoglobin, as we said, for oxygen transport. Myosin proteins generate force for muscle contraction. Insulin acts as a crucial signaling molecule regulating metabolism. And of course, thousands of different enzymes like hexokinase starting glycolysis or RNA polymerase transcribing genes, which catalyze nearly every chemical reaction needed for life.
So ultimately, the characteristics and traits of a whole organism are largely determined by the collection of proteins its cells can make.
To a very great extent, yes. It all comes back to having the right proteins in the right amounts at the right times, all folded correctly.
Okay, so we understand the code and the players, like tRNA and the importance of protein structure. But this code, it wasn't just, handed down on tablets. It had to be painstakingly figured out through some really clever experiments back in the day. This is where scientists really became detectives.
Indeed. The journey to actually crack the genetic code in the 1960s was a landmark period in molecular biology, a real scientific race in some ways. It really kicked off with the work of Marshall Nirenberg and J. Heinrich Matthaei.
What was their big breakthrough?
They developed what's called a cell-free translation system. Essentially, they took bacterial cells, broke them open, and isolated the key components needed for translation: ribosomes, tRNAs, enzymes, energy sources, all in a test tube.
So, like translation in a bottle.
Pretty much. If you added mRNA and amino acids to this mix, it would actually synthesize polypeptide chains.
Okay, that's cool. So they could control the input mRNA.
That was the key insight. They used an enzyme called polynucleotide phosphorylase, which was known to synthesize RNA strands randomly without needing a DNA template. Their first absolutely pivotal experiment involved creating an RNA strand made purely of uracil nucleotides. They called it poly-U.
Just UU.
Exactly. They added this poly-U to their cell-free system, along with all 20 amino acids, but only one type of amino acid was radioactively labeled in each separate tube.
To see which one got incorporated.
Precisely. After letting the system work, they precipitated any proteins made and checked for radioactivity, and they found that the poly-U directed the synthesis of a polypeptide made only of the amino acid phenylalanine.
Wow. So UUU must code for phenylalanine.
Bang. That was the first word deciphered. UUU equals phenylalanine. They quickly did the same with poly-A, finding it coded for lysine, poly-C, proline, and poly-G, glycine, though it was a bit tricky experimentally.
That's a huge start. But what about codons with mixed bases, like AUC or GCA? Poly-U wouldn't help there.
Right. For that, they started using mixed polymers. For example, if they made an RNA that was, say, 70% and 30% U, randomly mixed, they could predict the statistical frequency of different triplets. GGG would be most common, followed by GGU, GUG, DGG, then GU, UGU, UG, and finally UU would be least common. By measuring how much of each amino acid was incorporated, they could start inferring the composition of codons for those amino acids, even if they didn't know the exact sequence yet.
Clever, but still a bit indirect for the exact sequence.
It was. And that's where the incredibly elegant work of H. Gubin Khorana came in. He developed groundbreaking methods to chemically synthesize short DNA molecules with precisely repeating sequences. For instance, repeating dinucleotides like TCTR, or trinucleotides like TTC, or tetranucleotides.
Okay, synthetic DNA with known repeats.
Yes. These synthetic DNAs were then transcribed into RNA molecules that also had precisely repeating sequences. For example, an RNA like 5'-UCU-C3R contains only 2 alternating codons, UCU and CUC. When they put that RNA into the cell-free system,
It should make a protein with alternating amino acids.
exactly, and it did. They found poly-UC produced alternating serine, UCU, and leucine, CUC. By using different repeating di, tri, and tetranucleotide RNAs, Carena and his team could unambiguously assign many codons to their specific amino acids based on the repeating patterns of amino acids produced.
That's brilliant. Really nailing down the sequences. Was that enough to solve the whole code?
It got them most of the way there, but there's another ingenious technique that helped confirm assignments and fill in the last few gaps. This was Nirenberg and Philip Leder's triplet binding assay, developed around 1964.
Triplet binding. What did they do?
They discovered something quite remarkable. Even very short RNA molecules, just 3 nucleotides long, a synthetic triplet codon could direct the binding of the correct charged tRNA to a ribosome, even without actual translation happening.
So no polypeptide chain needed.
Nope. They would mix a known RNA triplet, say 5' CCC35, with ribosomes, and then add tRNAs charged with different amino acids, one of which was radioactively labeled. Only the tRNA whose anticodon matched the CCC triplet and was carrying the correct amino acid, proline in this case, would bind stably to the ribosome. They could then pass this mixture through a filter. The large ribosomes with the bound tRNA would get stuck, while unbound tRNAs passed through.
So if the filter was radioactive, they knew they had the right match.
Exactly. They tested all 64 possible triplets this way. It allowed them to directly and quickly determine the amino acids specified by each specific codon triplet. Between Nirenberg and Matthias polymers, Carena's repeating copolymers, and Nirenberg and Leder's triplet assay, the entire genetic code was deciphered. A monumental achievement, truly worthy of the Nobel Prizes awarded for it.
Absolutely incredible detective work. So we've cracked the code. But who are the key players actually doing the work of reading the mRNA and assembling the proteins? Let's properly meet the stars. tRNAs and ribosomes.
Right. We've touched on tRNA already, but let's look closer. Francis Crick, even before tRNA was fully characterized, proposed his famous adapter hypothesis. He reasoned there must be some molecule that could recognize the codons on the mRNA and also carry the corresponding amino acids.
The go-between molecule.
Precisely. And tRNA was later confirmed to be that adapter. As we said, it has that crucial dual role, recognizing the mRNA codon via its anticodon loop and carrying the correct amino acid attached to its other end, the acceptor stem.
And they have a distinctive shape.
Yes. When drawn flat, tRNA molecules typically fold into a cloverleaf secondary structure. This has several loops and stems. The anticodon is in one specific loop, ready to pair with the mRNA. The 3' end always ends with the sequence CCA, and that's where the amino acid gets attached. They also contain several modified bases, not just the standard A, U, G, C, which are important for their structure and function.
Okay, so the tRNA is the adapter, but it has to pick up the right amino acid first. How does the cell make sure the tRNA for, say, alanine actually gets loaded with alanine and not something else? That seems critical.
Absolutely critical. This is the job of a set of vital enzymes called aminoacyl tRNA synthetases. Think of them as the chargers for the tRNAs.
Okay.
There are 20 different synthetases in most cells, one dedicated to each of the 20 common amino acids. Each synthetase enzyme has 2 crucial recognition tasks. It must recognize its specific amino acid and must recognize all the correct tRNA molecules. Remember, degeneracy means there might be multiple tRNAs for one amino acid that are supposed to carry that amino acid.
How does it recognize the tRNA? Just the anticodon.
The anticodon is important, yes, but the synthetase often recognizes other features on the tRNA molecule as well, like parts of the acceptor stem or other loops. It's a highly specific molecular recognition process. The enzyme then uses the energy from ATP hydrolysis to covalently attach the correct amino acid to the 3' end of its matching tRNA.
And this has to be accurate.
Incredibly accurate. This charging step is sometimes called the second genetic code because its fidelity is just as important as correct codon-anticodon pairing during translation. Errors here, putting the wrong amino acid on a tRNA would directly lead to the wrong amino acid being inserted into proteins. Luckily, these synthetases are very precise and even have proofreading mechanisms to fix mistakes. The error rate is remarkably low, maybe less than 1 in 10,000.
Wow. Okay, that answers a question I had earlier. If there are 61 codons that code for amino acids, do we need 61 different types of tRNA molecules with unique anticodons? That still seems like a lot for the cell to make and manage.
That's an excellent question, and the answer is no. Cells typically don't need 61 different tRNAs. This is where another brilliant insight from Francis Crick comes in. The wobble hypothesis.
Wobble. Sounds a bit unstable.
Well, it refers to flexibility in pairing at the third position of the codon. Crick proposed that while the base pairing between the first two positions of the mRNA codon and the corresponding bases of the tRNA anticodon follows the standard strict rules, A with U, G with C, the pairing between the third base of the codon and the first base of the anticodon, they pair antiparallel, can be more relaxed or wobble.
Meaning non-standard pairs can form there.
Exactly. For example, a G in the wobble position of the anticodon can potentially pair with either C or U in the third position of the mRNA codon. Or inosine, a modified base often found in the anticodon wobble position, can pair with U, C, or A.
So one tRNA can actually recognize multiple codons.
Precisely. As long as those codons specify the same amino acid, which they usually do, thanks to the code's degeneracy, this wobble pairing means that a cell can translate all 61 sense codons using significantly fewer than 61 different tRNA types. E. coli, for instance, gets by with about 40 different tRNAs. It's another example of the cell's efficiency.
Very clever. Okay, so we have the charged tRNAs ready to go thanks to the synthetases and the wobble rules. Where does all the action actually happen? You mentioned the ribosome earlier, the macromolecular arena.
Yes, the ribosome. It's a truly magnificent molecular machine responsible for orchestrating the entire process of translation. Ribosomes are huge complexes made of ribosomal RNA, rRNA, which actually makes up most of their mass, and dozens of different ribosomal proteins.
And they differ between bacteria and eukaryotes.
They do, in size and composition, though their overall function is highly conserved. Bacterial ribosomes are called 70S ribosomes. They're made of a small 30S subunit containing 16S rRNA and a large 50S subunit containing 23S and 5S RNAs.
And ours, eukaryotic ones.
Eukaryotic ribosomes are larger, designated ADS. They consist of a small 40S subunit with 18S RNA and a large 60S subunit with 28S, 5.8S, and 5S RNAs. These size differences, the S stands for Svedberg units, a measure of sedimentation rate, not simple addition, are subtle but crucial, as we'll see later with antibiotics.
Where are they made?
In bacteria, assembly happens right in the cytoplasm. In eukaryotes, it's more complex. Ribosomal proteins are made in the cytoplasm, imported into the nucleus inside the nucleus, assembled with RNAs there, and then the completed subunits are exported back out to the cytoplasm to do their job.
Okay, so what does the ribosome actually do during translation? It must hold everything in place.
It does much more than just hold things. It has distinct functional sites that are essential for the process. There are 3 main sites where tRNAs bind, spanning across the small and large subunits.
The A, P, and E sites?
Exactly. The A site for aminosyl is where a new incoming charged tRNA first binds, matching the mRNA codon currently exposed there. The P site for peptidyl holds the tRNA carrying the growing polypeptide chain. And the E site for exit is where the uncharged tRNA sits briefly before leaving the ribosome after it has delivered its amino acid.
So the tRNAs move through these sites, A to P to E.
Precisely, in that order, as the ribosome moves along the mRNA. The ribosome facilitates the codon-anticodon recognition, catalyzes the peptide bond formation, and moves everything along. And often you'll see multiple ribosomes translating the same mRNA molecule simultaneously. This structure, like beads on a string, is called a polyribosome or polysomum.
Why do that?
Efficiency. It allows the cell to produce many copies of the same protein very rapidly from a single mRNA template.
Makes sense. Okay, so like any complex assembly process, translation must happen in defined stages. Like building a car, you have the chassis set up, then adding parts, then the final touches.
That's a perfect analogy. Translation proceeds in 3 main stages: initiation, elongation, and termination.
Let's start with initiation, getting things set up. How does that work in bacteria?
Okay, bacterial initiation involves several protein helpers called initiation factors, or IFs. First, IF1 and IF3 bind to the small 30S ribosomal subunit, which prevents the large 50S subunit from binding prematurely.
Keeping the subunits apart for now.
Right. Then, the mRNA binds to the 30S subunit. This binding is guided by a specific sequence in the mRNA's 5' untranslated region called the Schindel-Garneau sequence. It's typically just upstream of the AUG start codon and is complementary to a sequence in the 16S rRNA of the small subunit.
So the RNA itself helps position the mRNA correctly.
Exactly. It ensures the AUG start codon is positioned precisely in what will become the P site. Then another factor, IF2, bound to GTP, an energy molecule, escorts the special initiator tRNA to the start codon. In bacteria, this initiator tRNA carries a modified methionine called N-formylmethionine, tRNAF-MET.
Okay, initiator tRNA in place of the start codon in the P site.
Right. Once that's set, the initiation factors IF1 and IF3 are released. IF2 hydrolyzes its GTP, which provides energy, and it's also released. This allows the large 50S subunit to bind, completing the formation of the 70S initiation complex. Everything is now poised to start building polypeptide.
And how is initiation different in our cells and eukaryotes? You mentioned it's more complex.
It is, yeah. It involves many more initiation factors, called EIFs for eukaryotic. A key difference is that the eukaryotic initiator tRNA carries regular methionine, not the formulated version. Also, eukaryotic mRNAs don't have a Schindel-Garneau sequence.
So how does the ribosome find the start codon?
Instead, a group of EIFs, particularly one called EIF4, recognizes and binds to the unique 7-methylguanosine cap structure found at the very 5' end of eukaryotic mRNAs. This complex then recruits the small 40S ribosomal subunit along with the initiator tRNA already bound.
So it starts at the cap, then what?
Then this whole complex typically stands along the mRNA in the 5' to 3' direction, looking for the first AUG codon it encounters.
It slides along until it finds AUG.
Usually, yes. The efficiency of finding the right AUG is often influenced by the surrounding nucleotide sequence, described by what are called Kozak's rules. In optimal context, like having a purine A or G 3 bases before the AUG and a G immediately after it, helps signal this is the correct start.
Okay, it finds the right AUG.
Once the initiator tRNA's anticodon pairs with the start codon, other initiation factors are released, often involving GTP hydrolysis again, and the large 60S ribosomal subunit binds, forming the complete 80S initiation complex. The initiator tRNA is positioned in the P site, and the A site is empty, ready for the next step.
Got it. So once that whole initiation complex is assembled, the polypeptide chain starts to actually grow rapidly. This is the elongation phase, like the main assembly line clicking into gear.
Yes, and elongation is where the bulk of the protein synthesis happens. It's a cyclical process, repeating over and over for each amino acid added, and it's remarkably fast, especially in bacteria, maybe adding up to 20 amino acids every second.
Wow. What are the steps in one cycle?
Okay, 3 main steps per cycle. First, tRNA binding. A charged tRNA carrying the next amino acid specified by the codon currently sitting in the empty A site enters the A site. This binding is facilitated by an elongation factor protein, EF2 in bacteria, EFF1A in eukaryotes, which uses GTP hydrolysis for energy and accuracy checking.
Accuracy checking.
Yes, the ribosome has a decoding function, primarily involving the rRNA in the small subunit. It checks the codon-anticodon pairing. If it's not a correct match, the tRNA is rejected before the next step can happen. This is a major checkpoint for maintaining the high fidelity of translation, making sure the right amino acids are added.
Okay, so the correct tRNA is now in the A site. Step 2.
Step 2 is the crucial one, peptide bond formation. The ribosome catalyzes the transfer of the growing polypeptide chain from the tRNA in the P site onto the amino group of the amino acid attached to the tRNA in the A site. This forms a new peptide bond.
And this is done by the ribosome itself.
Yes.
Right.
And what's truly remarkable, as we hinted at earlier, is that this peptidyltransferase activity isn't performed by a ribosomal protein, but by the ribosomal RNA of the large subunit, 23S RNA in bacteria, 28S rRNA in eukaryotes. This discovery was huge. It showed that RNA itself can be catalytic, making the ribosome a ribozyme.
An RNA enzyme. Cool. So the chain is now 1 amino acid longer and attached to the tRNA in the A site. What's step 3?
Step 3 is translocation. The ribosome moves exactly 1 codon's length, 3 nucleotides, along the mRNA in the 3' direction. This movement requires another elongation factor, EFG in bacteria, EF2 in eukaryotes, and more GTP hydrolysis.
What does that movement achieve?
It shifts everything over. The tRNA that was in the A site, now carrying the longer polypeptide, moves into the P site. The tRNA that was in the P site, now uncharged, as it gave up the polypeptide, moves into the E site. And the A site is now empty again, exposing the next codon in the mRNA, ready for a new charged tRNA to bind.
And the uncharged tRNA in the E site.
It's released from the ribosome, and the cycle begins again. New tRNA binds to A site, peptide bond forms, ribosome translocates, over and over, adding amino acids one by one according to the mRNA sequence.
A beautiful, efficient cycle. But the polypeptide grows and grows.
Yeah.
It can't go on forever. There has to be a signal to stop building the chain.
Absolutely. That's the final stage, termination. It's triggered when the ribosome encounters one of the 3 stop codons, UAA, UAG, or UGA in the A site.
And you said earlier no tRNAs recognize these stop codons.
Correct. Instead, they are recognized by specific proteins called release factors, or RFs.
Release factors, what do they do?
They bind to the ribosome when a stop codon is in the A site. In bacteria, there are 2 main ones. RF1 recognizes UAA and UAG, while RF2 recognizes UAA and UGA. There's also an RF3 that assists. In eukaryotes, it's simpler. A single release factor, ERF1, recognizes all 3 stop codons, assisted by ERF3.
How do they bind there?
It's fascinating, really. Structurally, these release factors kind of mimic the shape of a tRNA molecule. This molecular mimicry allows them to fit precisely into the A site when a stop codon is present.
Clever. So the release factor binds. Then what?
Its binding triggers a crucial event: the hydrolysis, breaking with water, of the bond linking the completed polypeptide chain to the tRNA still sitting in the P site. This effectively cuts the finished protein loose from the translation machinery.
The protein is free.
Yes. And following that, the entire complex disassembles the ribosomal subunits separate from the mRNA, and the release factors and the last tRNA are released. Everything is then recycled, ready to start translating another mRNA molecule.
Initiation, elongation, termination. A complex dance, perfectly choreographed. You mentioned a key difference between bacteria and eukaryotes earlier, this coupling idea.
Yes, that's a really important distinction. Because bacteria don't have a nucleus separating their DNA from their ribosomes, transcription, making mRNA from DNA, and translation, making protein from mRNA, can happen simultaneously and in the same location.
So ribosomes can jump onto the mRNA while it's still being made.
Literally. As the 5' end of the mRNA emerges from the RNA polymerase enzyme that's busy transcribing the gene, ribosomes can latch on and start translating it immediately. You can see this under an electron microscope: multiple ribosomes translating an mRNA that's still attached to the DNA being transcribed. This is called coupling.
But they can't happen in our cells.
No, because in eukaryotes, transcription happens inside the nucleus, but translation happens out in the cytoplasm. The mRNA has to be fully transcribed, processed, capped, spliced, polyadenylated, and then exported from the nucleus before ribosomes in the cytoplasm can even access it. So transcription and translation are spatially and temporally separated. No coupling.
That makes sense. Now, connecting this back to something practical.
Yeah.
Why should someone, listening at home care about these intricate differences between bacterial ribosomes, 70S, and eukaryotic ribosomes, 80S? It sounds very academic.
But it's incredibly important for medicine. Those subtle structural and functional differences are precisely what many life-saving antibiotics exploit.
How so?
Many common antibiotics work by selectively inhibiting bacterial protein synthesis while leaving our eukaryotic ribosomes largely unharmed. They target aspects of the bacterial 70S ribosome or its function that are different from our 80S ribosomes.
Can you give some examples?
Sure. For instance, chloramphenicol binds to the 50S subunit and blocks the peptidyltransferase reaction, stops peptide bond formation. Erythromycin binds nearby and blocks the translocation step, jams the ribosome's movement. Tetracyclines interfere with the binding of charged tRNAs to the A site. Streptomycin actually causes the ribosome to misread the mRNA codons, leading to garbled proteins. And puromycin mimics a charged tRNA, gets incorporated briefly, but then causes the premature release of the unfinished polypeptide chain.
Wow, all targeting different steps of bacterial translation.
Exactly. And because our ADS ribosomes are different enough in structure, these drugs generally don't inhibit our own protein synthesis significantly at therapeutic doses. That specificity is what makes them effective antibacterial agents. It's a direct application of understanding these fundamental molecular differences.
That's a fantastic connection. So, reflecting on everything we've covered, from Garrett's really early insights into those inborn errors, through Beadle and Tatum figuring out the gene polypeptide link, the amazing race to decipher the genetic code, and now understanding the intricate molecular choreography of initiation, elongation, and termination. We've really taken a deep dive into one of life's most fundamental, essential processes.
We certainly have.
It's truly amazing when you stop and think about how meticulously the cell follows that genetic blueprint, building these absolutely essential protein machines second by second.
It really is staggering. And maybe, to leave you with something to think about, consider the sheer speed and generally incredible precision of translation despite all this complexity we've discussed. What might be the consequences if errors do occur, even rarely? How might mistakes in reading the code or building the protein impact our health? And conversely, as our understanding of these mechanisms gets even deeper, how might that continue to unlock totally new medical therapies, maybe ways to correct translational errors, or even design entirely new proteins or therapeutic RNAs? The possibilities are really quite profound.
That's a great thought to end on, the future possibilities based on understanding this core process. Well, thank you for joining us on this deep dive into the truly fascinating world of protein synthesis. We really appreciate you, our listeners, being a part of the Deep Dive family.